Transposable elements in eukaryotes, transposable elements, transposons in maize, P elements in Drosophila, Cynaealia family, line L1 of humans. Transposons have been discovered in eukaryotic organisms also for controlling elements in maize, PY elements in yeast, and P elements in Drosophila. These elements can be divided into two main classes. These classes include transposable elements that are similar to those found in bacteria. These elements contain inverted repeats at their ends and generate short direct repeats of the target DNA at the sites of their insertion. These elements are always located in the host genome and cannot survive outside the genome. Controlling elements in maize and P elements in Drosophila belong to this class of transposable elements. Retroposons, retrotransposons are DNA elements formed by the reverse transcription of retrovirus. This class of retrovirus and their sequences are transposed via RNA. Transposition of retroposons occur through RNA intermediate. Controlling elements in maize. McClintock experiments the DS element. In the 1940s, Barbara McClintock, during study of corn kernels, found that rather than being purple or white, they exhibit spots of purple pigment on normally white kernels. She knew that the phenotype was the result of an unstable mutation. From her careful genetic and cytological studies, she came to the conclusion that the spotted phenotype was not the result of any conventional kind of mutation, but rather was due to a controlling element which we now know as a transposon. According to Barbara McClintock, changes in maize genome during somatic cell division were genetically controlling abbreviations such as deficiencies, duplications, inversions, translocations, and ring chromosomes. Then dissociation activator, ACDA system. The explanation for the spotted kernels is that if the corn plant carries a wild type C gene, the kernel will be purple. Small c, colorless mutations block purple pigment production, so the kernel is colorless during kernel development. Reverts of the mutation occur, leading to a spot of purple pigment. The genetic nature of the reversion is supported by the fact that descendants of the cell which underwent the reversion also can produce the pigment. The earlier in development the reversion occurs, the larger is the purple spot. McClintock determined that the original C, colorless mutation, resulted from a mobile controlling element, a genetic factor called as DS, dissociation. This element gets inserted into the D gene. This action of DS is dependent on the presence of an unlinked gene, AS activator, or the changes were found to be caused by a genetic system named dissociation activator system. McClintock termed these genetic elements DS and AC as controlling elements in 1956. Since then, several systems of controlling elements have been discovered in maize. These elements are classified into two groups, autonomous and non-autonomous. Autonomous elements, the controlling elements which have the ability of their own excision and transposition, are called autonomous elements: activator, suppressor, mutator, and enhancer. Non-autonomous elements. These elements do not have the ability of transposition. Non-autonomous elements are originated from autonomous elements to the loss of transacting functions which are required for transposition. A single type of autonomous element and a different non-autonomous elements derived from it form a family. Deletions of different lengths and different regions from an autonomous element give rise to different types of non-autonomous elements. Such elements change their position in response to an autonomous element of the same family present in the genome. Deletions of different lengths and different regions from an autonomous element give rise to different types of non-autonomous elements. Such elements change their position in response to an autonomous element of the same family present in the genome. Non-autonomous element is activated in trans by its related autonomous element. Examples of non-autonomous elements are dissociation, defective suppressor mutation, and inhibitor. AC is required for transposition of DS into a G. AC can also move the DS out of the C gene, resulting in the wild type revertant, a purple spot. McClintock found it impossible to map AC. In some plants, it mapped to one position. The other plants of the same line, it mapped to different positions. Moreover, the DC locus itself was constantly changing positions on the chromosome arm, as indicated by the different phenotypes of the varigard sections of the seeds. AC is autonomous element of the family, and hence mutations caused by AC are unstable. DC is the non-autonomous element of the family. DC mutations are stable if only DC are present. They are unstable in the presence of an AC element. The main occurrence of a DC system in maize as follows. The AC element can exist in a number of states similar to other genes. It controls the activity and time of action of the DS element. AC and DC both exhibit interchromosomal as well as intrachromosomal movements or transposition, and the movement occurs through excision of these elements from one site and their insertion at a new site. DS element is unstable in the presence of AC element in the same nucleus. When both the elements are present, loss deletion of a part of chromosome 9 occurs if the chromosome 9 carries the DS element. The deletion is caused by breakage of the chromosome at the site of DS. The gene lying adjacent to DS becomes inactivated. AC with 11 base pair imperfect terminal inverted repeats IRs. AC contains a single transcription unit that comprises most of the element's length. DC does not transpose in the absence of AC and maintains as a stable insertion in the chromosome. When an AC element is present, it activates DS, causing it to transpose to a new site or to break the chromosome in which it is located. DC elements are heterogeneous in length and sequence. All DC elements have the same terminal IRs and AC elements, and many of these DS elements have been generated from AC by deletion of various lengths. AC exhibits conservative transposition. AC transpose only during replication. Purple kernels AC, DC, C, normal C expressing pigment product. Colorless kernel activated DC transposition. D can transpose to C. So spotted kernels, disrupted C gene mutant, spotted kernel. Normal gene change in color in corn due to transposition effect. General characteristics of controlling elements. Several systems like DCSAC have now been found in corn. This shows similar action, having a target gene that is inactivated presumably by the insertion of some receptor element into it, and a distant regulator gene that maintains the mutational instability of the locus, presumably through its ability to unlock the receptor element from the target locus and return the locus to normal function. The receptor and the regulator are termed controlling elements. In the example considered so far, the unstable allele is said to be non-autonomous. It can revert only in the presence of the regulator. Sometimes, however, a system such as ACDS system can produce an unstable allele that is autonomous. Such mutants are recognizable because they show Mendelian ratios such as 3:1 for pigmented to dotted that apparently are independent of any other element. In fact, such alleles appear to be caused by the insertion of AC itself into the target gene. An allele of this type can subsequently be transformed into a non-autonomous allele. In such case, the non-autonomous seems to result from the spontaneous generation of a DS element from the inserted AC element. In other words, DS is in all likelihood an incomplete version of AC itself. Next topic, transposable genetic elements in prokaryotes. The four transposable genetic elements in prokaryotes are bacterial insertion sequence, prokaryotic transposons, insertion sequence elements, and transposons in plasmids, and phage MU. First, bacterial insertion sequences. Insertion sequences or insertion sequence elements, IS elements. Insertion sequence or insertion sequence elements are now known to be segments of bacterial DNA that can move from one position on a chromosome to a different position on the same chromosome or on different chromosome. An IS element contains only genes required for mobilizing the element and inserting the element into chromosome at a new location. IS element are normal constituents of bacterial chromosome and plasmid. When IS element appear in the middle of genes, they interrupt the coding sequence and inactivate the expression of that gene. Owing to their size and in some cases the presence of transcription and translation termination signals, IS elements can also block the expression of other genes in the same operon if those genes are downstream from the promoter of the operon. IS elements were first found in E. coli as a result of their effect on the expression of a set of 3 genes whose products are needed to metabolize a sugar galactose as a carbon source. Careful investigation showed that the mutant phenotypes resulted from the insertion of approximately 80 base pairs DNA segment into a gene. This particular DNA segment is now called insertion sequential. Next, properties of IS element. IS1 is a genetic element capable of moving around the genome. It integrates into the chromosome at locations with which it has no homologue, thereby distinguishing it from recombination. This event is an example of transposition event. There are now number of IS elements that have been identified in E. coli, including 1s1, IS1, IS2, IS10, which present on 0 to 30 copies per genome and each with a characteristic length and unique nucleotide sequence. IS1 is 7868 base pair long and is present in 4 to 19 copies on the E. coli chromosomes. IS1 is present in 0 to 12 copies on the E. coli chromosome and in 1 copy on the F plasmid. And IS10 is found in a class of plasmids called R plasmid that can replicate in E. coli. Among prokaryotes, the IS elements are normal cell constituents, that is, they are found in most cells. Altogether, IS elements constitute approximately 0.3% of cell's genome. All IS elements that have been sequenced end with perfect or nearly perfect inverted terminal repeats of between 9 and 41 base pairs. This means that essentially the same sequence is found at each end of a IS but in opposite orientation. IS element picture structure of a bacterial IS element. 5 to 11 base pair direct repeat protein coding region. 50 base pair inverted repeat IS element protein coding region. Structure of bacterial IS element. Then transposition, IS transposition. When transposition of an IS element takes place, a copy of the IS element inserts into a new chromosome location while the original IS element remains in place. That is, transposition requires the precise replication of the original IS element using the replication enzymes of the host cell. The actual transposition also requires an enzyme encoded by the IS element called transposase. The IR sequences are essential for the transposition process. That is, those sequences are recognized by transposase to initiate transposition. IS elements insert into the chromosomes at sites with which they have no sequence homologue. Genetic recombination between non-homologous sequences is called illegitimate recombination. The sites into which IS elements insert are called target sites. The process of IS insertion into a chromosome is important. Firstly, a tagged cut is made in the target site and IS element is then inserted, becoming joined to the single stranded ends. The gaps are filled in by DNA polymerase and DNA ligands, producing an integrated IS element with 2 direct repeats of the target site sequence flanking the IS element. Direct in this case means that the 2 sequences are repeated in the same orientation. The direct repeats are called target site duplications. The sizes of target size duplication vary with the IS element but tend to be small. Integration of some IS elements show preferences for certain regions while others integrate only at particular sequences. All copies of a given IS element have the same sequence, including that of the inverted terminal repeats. Mutations that affect the inverted terminal repeat sequence of IS elements affect transposition, indicating that inverted terminal repeat sequence are the key sequences recognized by transposases during a transposition event. So, insertion of IS element into chromosomal DNA. Insertion IS element, sticky ends of host DNA, new DNA, gap filled by DNA polymerase, nick sealed by ligase, schematic representation of the integration of an IS element into chromosomal DNA. Next, prokaryotic transposons. A transposon or TN is more complex than an IS element. A transposon is a mobile DNA segment that, like an IS element, contains genes for the insertion of DNA segment into a chromosome and for the mobilization of the element to other locations on the chromosome. There are two types of prokaryotic transposons: composite transposons and non-composite transposons. Composite transposons, they are complex transposons with a central region containing gene. Drug resistance genes flanked on both sides of IS element, also called IS modules. Composite transposons may be thousands of base pairs long. The IS elements are both of the same type and are called ISL for left and ISR for right. Depending upon the transposon, ISL and ISR may be in the same or inverted orientation relative to each other. Because the ISS themselves have terminal inverted repeats, the composite transposons also have terminal inverted repeats. Structure of composition transposon TN10. To illustrate the general features of such transposons, the TN10 transposon is long and consists of non-repeating DNA containing a tetracycline resistant gene flanked at each end with IS element. This IS element are designated IS10L and IS10R and are arranged in the inverted orientation. Cells containing TN10 are resistant to tetracycline resistance gene contained within the central DNA sequence. Transposition of composite transposons occur because of the functions of IS elements they contain. One or both IS elements supplies the transposase. The inverted repeats of the IS elements at the two ends of the transposon are organized by transposase to initiate a transposition. As with transposition of IS elements, transposition of TN10 is rare, occurring once in 10 cell generations. This is the case because less than one transposase molecule per cell generation is made by TN10. Like IS elements, composite transposons produce target site duplications after transposition. Inverted repeat of IS element, inverted repeat of IS element, tetracycline resistant, invert IS. Just detailed structure of a TN10 transposon. Then non-composite transposons. They, like composite transposons, contain genes which are those for drug resistance. Unlike composite transposons, they do not terminate with IS element. They do not have the repeated sequence at their ends that are required for transposition. TN3 is a non-composite transposon. TN3 has a 38 base pair inverted terminal repeat and contains 3 genes in its central region. One of those genes, DLA, encodes beta lactamase, which breaks down ampicillin and therefore makes cells containing TN3 resistant to ampicillin. The other two genes, TNPA and TNPB, encode the enzyme transposase and resolvase that are needed for transposition of TN3. Transposase catalyzes insertion of the TN into new sites, and resolvase is an enzyme involved in a particular recombination event associated with the transposition. Resolvase is not found in all transposons. The genes for transposition are in the central region for non-composite transposons, while they are in the terminal IS element for composite transposons. Non-composite transposons also cause target site duplications when they move. The transposons TN3 left inverted repeat, transposase, resolvase, right inverted repeat. Detail the structure of TN3 transposon. Then mechanism of transposition in prokaryotes. Several different mechanisms of transpositions are employed by prokaryotic transposable elements, and as we see later, eukaryotic elements exhibit still additional mechanisms of transposition. In E. coli, we can identify replicative and conservative non-replicative modes of transposition. In the replicative pathway, a new copy of transposable element is generated in the transposition event. The results of the transposition are that the one copy appears at the new site and the new copy remains at the old site. In the conservative pathway, there is no replication. Instead, the element is excited from the chromosome or plasmid and is integrated into the new site. Conservative mode of transposition, replicative mode of transposition. Then replicative transposition. The transposition of TN3 occurs in two stages. Firstly, the transposase mediates the fusion of two molecules forming a structure called a cointegrate. During this process, the transposon is replicated and one copy is inserted at each junction In the co-integrate, the two T and 3 are oriented in the same direction. In the second stage of transposition, the TNPR encoded resolvase mediates a site-specific recombination event between the two TN3 elements. This event occurs at a sequence in TN3 called RUS, the resolution site, and generates two molecules, each with a copy of the transposon. The TNPR gene product also has another function, namely to repress the synthesis of both the transposase and resolvase proteins. This repression occurs because the RUS site is located in between the TNPA and TNPR genes. By binding to this site, the TNPR protein interferes with the synthesis of both the gene products, leaving them in chronic short supply. Consequently, the TN3 element tends to remain immobile. So plasmid with TN3 target plasmid co-integrate, a resolution of co-integrate, resolvase, original transposition products, new. Then reservative transposition. Some transposons such as TN10 excise from the chromosome and integrate into the target DNA. In these cases, DNA replication of the element does not occur and the element is lost from the site of the original chromosome. This mechanism is called conservative non-replicative transposition or simple insertion. TN10 transposes by conservative transposition. Insertion of a transposon into a reading frame of a gene will disrupt it, causing a loss of function of that gene. Insertion into gene's controlling region can cause changes in the level of expression of the gene. Deletion and insertion events also occur as a result of activities of transposons and from crossing over between duplicated transposons in the genome. Then IS elements and transposons in plasmids. The transfer of genetic material between conjugating E. coli is the result of a function of the fertility factor F. DEF factor, a circular double-stranded DNA molecule, is one of the examples of bacterial plasmid. Plasmids such as F that are also capable of integrating into the bacterial chromosomes are called episomes. F factor that code for a variety of proteins. The important elements are transfer gene required for the conjugation transfer of the DNA, genes that encode proteins required for plasmid replication, 4 IS elements, 2 copies of IS3, 1 of IS2 and 1 of insertion sequence element called gamma delta. It is because the E. coli chromosome has copies of these 4 insertion sequences at various positions that the F factor can integrate into the E. coli chromosome at different sites and in different orientations with homologous sequence of the insertion elements. And the class of plasmids that has medical significance is the R plasmid group, which was discovered in Japan in the 1950s during the cure for dysentery. The disease is the result of infection by the pathogenic bacterium Shigella was found to be resistant to most of the commonly used antibiotics. Consequently, they found that the genes responsible for the drug resistance were carried on R plasmids, which can promote the transfer of genes between bacteria by conjugation, just as the F factor. One segment of an R plasmid is that homologous to a segment, and the F factor is the part needed for the conjugal transfer of genes. That segment and the plasmid specific genes for DNA replication constitute what is called the RTF, resistance transfer factor. The rest of the R plasmid differs from type to type and include the antibiotic resistant genes or other types of genes of medical significance, such as resistance to heavy metal ions. Plasmids. The resistance genes in our plasmid are in fact transposons. That is, each resistance gene is located between flanking indirectly repeated segments such as ones in IS modules. Thus, each transposon of the resistance gene in our plasmid can be inserted into new locations on other plasmids or on the bacterial chromosome, while at the same time leaving behind a copy of itself in the original position. So, chromosomal resistance, transposon, tetracycline resistance. Two different transposons having different inverted repeats regions and carrying different drug resistant genes. TN9 has a short IR region because the two IS1 elements are in the same orientation and each element has a short inverted repeat. TN10 has a large IR region because the two IS10 components have opposite orientation and the entire IS10 sequence constitutes the inverted repeat. Then phage MU stands for mutator. Phage MU is a normal appearing phage. We consider it here because although it is a true virus, it has many features in common with IS elements. The DNA double helix of this phage is 36,000 nucleotides long, much larger than an IS element. However, it does appear to be able to insert itself anywhere in a bacterial or plasmid genome in either orientation. Once inserted, it causes mutations at the locus of insertion, again like an IS element. The phage was named for its ability. Mu stands for mutator. Normally, these mutations cannot be reversed, but reversion can be produced by certain kinds of genetic manipulation. When this reversion is produced, the phages that can be recovered showing no deletion, proving that excision is exact and that the insertion of the phage therefore does not involve any loss of phage material either. Each mature phage particle has on each end a piece of flanking DNA from its previous host. However, this DNA is not inserted anew into the next host. This function is unclear. Phage mu also has an IR sequence, but neither of the repeated elements is at a terminus. So host DNA tail, mu genome. The DNA at the free mu phage has reversed from its previous host. Mu can also act as a genetic snap faster, mobilizing any kind of DNA and transposing it anywhere in a genome. For example, it can mobilize another phage such as lambda or the F factor. In such situations, the inserted DNA is flanked by two mu genomes, host DNA tail, mu genome, lambda genome, mu genome, and host DNA tail. Phage mu can mediate the insertion of phage lambda into bacterial chromosomes, resulting in structure like one shown here. Another explanation, transposable elements, meaning, nomenclature, and classes. In this article, we will discuss about meaning of transposable elements, nomenclature of transposable elements, classes, and mechanism. The term transposon was coined by Hedges and Jacob in 1974 for mobile discrete DNA sequences in the genome. Such sequences are able to transport themselves from one location to the other location within the genome. These sequences are also called transposable genetic elements. Transposable genetic elements are found in both prokaryotes and eukaryotes. Each bacterial transposon carries gene coding for the enzyme needed for its own transposition. Similar systems occur in eukaryotic organisms also. Transposons promote rearrangements of the genome in two ways. Transposition causes depletion or inversion, and transposable elements act as regions of homology or recombination, resulting in translocations, inversions, deletions, and insertions. So, meaning of transposable elements. For a long time, it was assumed that the genes have fixed chromosomal locations. Genetic recombination is a common process of itself within which exchange between the homologous DNA sequences take place. Over the past few decades, some information of gene arrangement have come forth that result from illegitimate crossing over between incomplete homologous DNA sequences. In 1940, McClintock Barbara, in her genetic experiment with maize, found that certain genetic elements regularly jump to new locations and thereby affect gene expression. Therefore, kernels of maize ears show variations in color. Her work was followed even after 30 years by the recognition that bacteria contain mobile DNA sequence. The genetic segments moving from one location to the other were also discovered in bacteria well during 1970s, but the frequency was quite low. Several apparent random mutations disrupting gene function in E. coli were found to occur due to insertion of a large DNA segment. Many of these sequences had the jumping properties similar to maize elements. Later on, the properties of such genes, E. coli, could be known at molecular levels through the methods of gene cloning and DNA sequencing. These moving genetic elements have been called with different names such as jumping genes, movable genes, transposons, and transposable elements. For consistent work on maize and discovery of jumping genes, McClintock was awarded Nobel Prize in 1983. Genome evolving by new sequences and by rearrangement of existing sequence. The sudden introduction of new sequences results from the ability of a factor to carry information between gene loci. The extra chromosomal element provides informations by transferring a small DNA segment. In bacteria, plasmids move through conjugation, whereas phages transfer through infection. Both phages and plasmids occasionally transfer host genes along with their own replication. In some bacteria, direct transfer of DNA occurs by transformation. In addition, the retrovirus can transfer DNA segment during infection cycle in eukaryotes. However, gene arrangement in genome may form new sequences by placing them in a new regulatory situation. Rearrangement occurs either from recombination undertaken by the cellular system for homologous recombination and repair or for the transportation induced by transposons. Movement of transposable elements from one chromosome site to another is shown. Movement of transposable element from one chromosome site to another. Arrows indicate the sites for action of enzymes and cutting of DNA strands. Chromosome A transposable element, chromosome B intermediate DNA rejoins. Generally, the mobile segment of template phage like lambda and mu phages, which resemble insertion sequences, are called transposable element, and the mobile sequence of bacterial system is called transposons. Thus, the transposable elements have been defined as sequences which can insert into several sites in a genome. Transposable elements are recognized by the presence of inverted repeat DNA sequence at their needs. These sequences are necessary for the DNA between them to be transported by a particular enzyme or transposase associated with the transposable element. The nomenclature of transposable elements, Campbell in 1977 have described the nomenclature of transposable elements in prokaryotes. The transposons, which were discovered first, did not contain any non-host genes. Therefore, the historical reasons these were called insertion sequences or IS elements, as designed as IS1, IS2, IS3, etc. So, transposons contain easily recognizable bacterial genes, especially for antibiotic resistance. The members of the class are designated by abbreviation TN followed by numbers such as TN1, TN2, TN3, etc. The number distinguishes different transposons when it is necessary to refer the genes carried on transposons. These are represented by standard genotypical designations such as TN1, where TN AMP refers that the transposons carries the genetic locus for ampicillin resistance. Moreover, transposon present within a particular gene creates mutation in that gene. For example, if mutation occurs at position 135 in GAL T gene by transposon 4, the notation is designated as GAL T 135 TN4. Transposons have occasionally been designated in non-standard ways also. For example, gamma and delta, which is an element present in F plasmid. However, in eukaryotes, no definite pattern of naming of transposable element is used. For example, the transposable element Drosophila are named Copia 4, 9, 7, and P. Transposable element of yeast is TYL, and transposable element of maize as dissociation element and activator. Next, classes of transposable elements. Transposable elements of prokaryotes are generally called transposons. Mainly three classes of transposable elements have been categorized. Insertion sequence or IS elements, also called single transposons, which contain no genetic information except the DNA sequence necessary for transposition and also lack host gene. Transposons, which contain antibiotic resistance genes and may or may not be flanked by two identical copies of IS elements, and the transposable phages that are lysogenic phages and employ transposition as a way of life. Then insertion sequence, IS elements. For the first time, transposable elements were identified in the form of spontaneous insertion in bacterial operons. Such insertions inhibited the transcription and or translation of genes in which it was inserted. These simplest transposable elements were called insertion sequences and designated by the prefix IS, followed by members of each type. The IS elements are the normal constituents of bacterial chromosomes and plasmids. A typical strain of E. coli contains more than 10 copies of at least one of the common IS elements. The properties of some IS elements are given. The IS elements are the autonomous units of each, which codes only for proteins required for its own transposition. Each IS element differs in sequence size but resemble in several ways in organization. Mostly all of them are of about 1000 BP long, ending with inverted terminal repeats of about 10 to 40 base pair long. The inverted terminal repeat has two copies of repeats which are closely related. For example, the same sequence of inverted terminal repeat of 9 base pair is encountered from the flanking DNA on either side of it after proceeding towards the element. The terminal repeats are believed to serve as recognition sequence for transposition enzymes. The transposons in their role of fusing the ends of IS elements with the target DNA. So besides IS1, all the IS elements possess a single long coding sequence that starts just inside at one end of ITR and terminates just before or after inverted repeat at the other end. This codes for the transposase. However, IS1 consists of a more complex organization with 2 reading frames. The transposase is produced by making a frame shift translation to allow both reading frames to be used. After transposition by IS element, a sequence of target site of host DNA at the site of insertion is duplicated. The duplicated segment is known as direct repeat. It is repeated by the same orientation. This duplication of sequence of target site is revealed upon compares before and after insertion of IS element. Before after composition, one copy of sequence of target direct repeat is present in both the ends of transposon. The insertion sequence can be detected in 2 ways. They interact and inactivate genes into which they insert, and they may contain promoters which allow RNA polymerase to transcribe and thus turn on adjacent genes. They do not perform any function in bacterial cells except that they may act as natural agent of genetic change by bringing about structure and function. Many structural variants have been observed in IS element regarding the source and host range of IS elements. 2 generalizations can be made. The same kind of IS elements as IS1 can be found on plasmid, phage, genome, and chromosome of different bacteria. Several IS elements can function efficiently in bacteria that do not contain the same IS element in their chromosome. For example, IS10 is active in both E. coli K12 and Salmonella typhimurium, although their chromosome do not contain IS10 sequence. Several copies of IS elements may be found in bacterial genome. For example, 8 copies of IS1 and above 5 copies of IS2 are found in E. coli chromosomes. Then mechanism of transposition. Transcription of the gene producing transposase is regulated by a repressor. The repressor also regulates its own synthesis. The transposase produces a staggered cleavage at the target site in the host DNA in which the TN element is to transpose. The staggered cuts enable the joining of the single stranded ends of the host DNA with the ends of the transposon. After the joining of the ends, a single stranded open target genes sequences at the ends of the transposon support DNA synthesis leads to gap filling. Then the ligation of the next causes the duplication of the target sequence that make a direct repeat at both the ends of the inserted transposon. The mechanism of transposition are of the following 3 types. Then replicative, non-replicative, and conservative. So replicative transposition, the transposon is duplicated during the process of transposition. One copy of the TN element remains at the original site while the duplicated copy is inserted at a new site. Such type of transposition is a characteristic of DNA group of transposons. In this type of transposition, the number of copies of the transposon increase. 2 endonucleases are involved in transposition. The enzyme transposase acts on the ends of original transposition while another enzyme resolves acts on the duplicated copy. Non-duplicative transposition or conservative transposition. In this case, the transposable element moves from one site into the another. Since there is no duplication of the transposon, it is lost from the original site. This process involves a single enzyme, the transposon. Transposition of all IS elements and of the composite transposons TN5 and TN10 occurs according to this mechanism. This process is quite similar to the integration of phage lambda into E. coli chromosome. In such transposon, the transposons belong to lambda integrase family. Large transposon utilize the conservative mechanism of transposition. Next, phage MU. Phage MU is a normal appearing phage. We consider it here because although it is a true virus, it has many features in common with IS elements. With the DNA double helix of this phage is 36,000 nucleotides long, much larger than IS element. However, it does appear to be able to insert itself anywhere in a bacterial or plasmid genome in either orientation. Once inserted, it causes mutations at the locus of insertion, again like an IS element. The phage was named for this ability, mu transfer mutator. Normally, these mutations cannot be reverted, but reversion can be produced by certain kinds of genetic manipulation. When this reversion is produced, the phages that can be recovered showing no deletion, proving that excision is exact and that the insertion of the phage therefore does not involve any loss of phage material either. Each mutator phage particle has on each end a piece of flanking DNA from its previous host. However, this DNA is not inserted anew into the new host. Its function is unclear. Phage mu also has an IR sequence, but neither of the repeated element is at a terminus. Host DNA tail mu genome. The DNA of a free mu phage has tails derived from its previous host. Mu transposon contain host DNA tail. Phage mu can mediate the insertion of phage lambda into a bacterial chromosome, resulting in a structure like the one shown here. Host DNA tail, mu genome, lambda genome, mu genome, and host DNA tail will be there. So mu transposon, the bacterial phage mu is very large and is regarded as a giant mutator DNA element. It possesses a number of genes for its head and tail formation. This transposon has the ability to insert into E. coli chromosome at different non-homologous positions. The enzyme mu transposase cuts both the ends of transposon from the donor molecule. A staggered cleavage of 5 bases occur at the target site of insertion. Mu utilizes both the replicative and non-replicative pathways of transposition. This integration into the host genome occurs through non-replicative process, while the multiplication of mu occurs through the replicative mechanism. The mu transposon can produce 100 copies of its transposition to 100 different target sites in a single E. coli chromosome. The enzyme mu transposase organizes the terminal sequence and makes staggered cleavages at the end of the mu and also at the target sites in the chromosomal DNA. One cut end of the mu joins with the cut end of the target site of the recipient chromosome, producing a cross-shaped structure. Subsequently, one of the following two pathways is followed: transposition, donor molecule, target, recipient, staggered cleavage in transposon and target sites. Cut ends of transposon join to cut ends of the target DNA, then crossover structure, cut in both ends of donor strand, non-replicative, replicative. Transposon inserted into target DNA, donor with double-stranded break. So, co-integrate is also there. Replication from 2,3' ends to produce two copies of the transposition in a single co-integrate module. Arrows indicate the direction of DNA replication. So, non-replication and replicative mechanisms of transposition of mu. Both mechanisms have an initial common crossover structure. The non-replicative transposition results in a double-stranded break in the donor molecule. In replicative mechanisms, transposon is doubled and co-integrate is produced. A cleavage in the donor molecule near the transposition causes the insertion of the transposon in a target DNA. The single-stranded region formed by the staggered cleavage is filled by repair synthesis. This is the non-replicative mechanisms in which donor DNA suffers a double-stranded break and loses the mu transposon. So in the implicative mechanism, no further cleavage occurs in the cross-shaped structure. DNA synthesis occurs complementary to the single-stranded region that was formed by aggregated cleavage. Synthesis continues through the transposon and separates the duplex into single large DNA molecule consisting of both the donor and recipient chromosomes. The molecule containing two copies of transposon is called co-integrate. Transposons are located joining the donor and the recipient replicants. Rearrangement of host DNA caused by transposons. Transposons may often bring about changes in the host DNA as they have caused deletions, duplications, and inversions. When a copy of transposition is mobilized and inserted into the same chromosome near its original site, recombination may occur between the two copies. There are two situations depending on whether the orientation of the repeats is direct, inverted, direct repeats. In case of TN9, IS1 is present on both the ends as direct repeats. Recombination between a pair of direct repeats produces a circular DNA molecule that contains only one copy of the direct repeat. The other copy of the direct repeat remains within the chromosome. A circular DNA molecule is lost from the cell and thus a deletion occurs. So host chromosome is there, IS1, direct repeats, IS1, host chromosome, pairing and closing over between direct repeats, host chromosome, TN9 lacking one IS1 model, circular DNA it is lost. Recombination between the terminal direct repeats leads to deletion of the material between them. So TN9 minus 1 IS1 module is lost as a circular DNA, leaving an IS1 copy in the host chromosome. Then inverted repeats. The transposon TN903 consists of inverted repeats of IS909, 9093 at its two ends. A recombination between a pair of inverted repeats does not cause a deletion, but it results in an inversion of the central region. So inverted repeats host DNA IS903, IN903. Pairing and crossing over between the terminal inverted repeats. Recombination between inverted repeats of a composite transposon leads to inversion of the region between them. Here the original gene order ABCD is reverted to yield DCBA after the recombination event. Please look at this figure. Like this after recombination between inverted repeats of composite transposon that lead to inversion of the region between them. Next topic, transposable elements in eukaryotes. There are many different types of transposons in eukaryotes which vary in size, structure, composition and behavior. All the eukaryotic transposons have basic characteristics such as having terminal inverted repeats and target specific duplication when inserted. So types of transposons in eukaryotes. Class 1 retrotransposons. Example, TY elements in yeast, copia elements in Drosophila, line enzyme sequences in humans. So class 2 DNA transposons. Example, P elements in Drosophila. These are cut and paste transposons similar to bacterial transposons. Donor DNA retrotransposon. Class 1. Transcription, RNA intermediate, reverse transcription, DNA intermediate, integration, transposable element, target DNA. Then next, DNA transposon, class 2, donor DNA, excision, DNA intermediate, target DNA, integration, transposable element, target DNA on the other sides. So class 1, retrotransposons. Retrotransposons are those transposable elements that utilize reverse transcriptase enzyme to convert RNA intermediate into DNA and then transpose into new position by itself. They are of two types on the basis of long terminal repeats or LTR. LTR retrotransposons are retroviruses-like element. All LTR retrotransposons have the same basic structure, a central coding region flanked by long terminal repeats LTR which are oriented in the same direction. The LTR are typically a few hundred nucleotide pairs long sequence having short inverted repeats at both ends like present in other types of transposons. Due to characteristic LTR sequence which is similar to that of retrovirus, these transposons are known as LTR retrotransposons. The property of LTR transposons is similar to retrovirus infection. LTR retrotransposon utilizes reverse transcriptase enzyme from retrovirus that convert transposons RNA into DNA in the first state, and in the second stage the synthesized DNA is transported into new locations in the chromosome. So TY elements in yeast and copia elements of Drosophila, for example. First, TY elements in yeast. TY elements in is it LTR retrotransposon present in yeast, which is about 5.9 kilobase pair long sequence having LTRs of about 340 base pairs long at both ends. Most yeast strains have about 35 copies of TY1 element in their genome. TY1 elements have only two genes, TYE and TYB. which are homologous to the GAG and pol genes of the retroviruses. Transposition of TY1 element involves reverse transcriptase enzyme. After DNA is synthesized from TY1 DNA, reverse transcriptase encoded by the TYB gene uses it as a template to make double-stranded DNA. And then the newly synthesized DNA is transported to the nucleus and inserted in targeted site in the genome, creating a new TY1 element. Transposition in the yeast TY1 element. Structural protein, reverse transcriptase enzyme, TYA gene, TYB gene, LTR. So the TY1 element is transcribed to produce RNA. The RNA is reverse transcribed into DNA by an enzyme encoded by the TYB gene. The DNA is inserted into a chromosome, creating a new copy of the TY1 element. Non-LTR retrotransposons or retroposons. The non-LTR retrotransposons are common retrotransposons present in eukaryotes. They are simply known as retroposons or non-viral transposons. Non-LTR retrotransposons lack long terminal repeats or LTR in the terminal end. However, they have homologous sequences of 80 base pairs at one end, which is divided from post-transcriptional modifications, poly-A-tail. These non-LTR retrotransposons are transposed via RNA intermediate, which is reversed transcribed into double-stranded DNA by reverse transcriptase enzyme encoded by the element itself and then transposed to the target site. So transposable element of human, line, long interspaced nuclear elements, sign, short interspaced nuclear element, then transposable element of human. At least 44% of human genome is repetitive, which is derived from transposable elements, including retrovirus-like elements, retroposons, and several families of elements that transport by a cut and paste mechanism. Class of human transposons, line and sign. Long interspaced nuclear elements or lines. L1 transposon is the principal human transposable element which belongs to long interspaced nuclear elements class. Two other line sequences, L2 and L3 lines, are also present in human genome. However, both are transpositionally inactive. Complete L1 element is about 6 kb long sequence with an internal promoter that is recognized by RNA polymerase II and two open reading frames, ORFs. ORF1 encoded nucleic acid binding protein. ORF2 encode a protein with endonuclease and reverse transcriptase activities. The human genome contains about 3000 to 5000 copies of complete L1 elements, and this picture depicts this process. In short, just have a look at this. If you need to read this, just pause the video. A complete L1 element inserted in a chromosome, transcribed into L1 RNA. The L1 RNA is polyadenylated in the nucleus. Transposition of L1 element involves transcription of a complete L1 element into RNA in the first step and the reverse transcription of this RNA into DNA in next step. Both the transcription and reverse transcription process take place in the nucleus. However, before the L1 RNA is reverse transcribed, it travels to the cytoplasm where it is translated into polypeptides, nucleic acid binding proteins, endonucleases, and reverse transcriptase. The polypeptide remain attached with RNA when it returns to the nucleus. The polypeptide encoded by ORF2 possesses an endonuclease activity that catalyzes cleavage of one strand of the DNA duplex at targeted site in a chromosome. The poly-A tail of polypeptide is juxtaposed with 5' end of a cleaved DNA, and the reverse transcriptase activity results in the synthesis of single DNA strand. Next, utilizing 3' end as a primer for DNA synthesis, a newly synthesized L1 DNA is subsequently made double-stranded. Therefore, new L1 element is placed in new location. So, short interspaced nuclear element, sign. The short interspaced nuclear element are the second most abundant class of transposable elements in the human genome. Signs are less than 400 base pairs long and have an internal promoter, but do not encode protein. Like all retroposons, signs do not have terminal repeats, but instead they have a sequence of 8D base pairs at one end. Transposition of sign involves transcription of a sign DNA into RNA, which is then reverse transcribed into DNA. However, sign do not have any open reading frame to encode any enzyme. Therefore, it depends upon lines type element for reverse transcriptase enzyme and other enzymes. This signs depends on line to multiply and insert within the genome. These three families of signs are present in human genome: ALU, MIR, and TER2 and MI are three elements. However, only the ALU elements is transpositionally active. Class 2 DNA transposons. Some mobile elements found in eukaryotes are DNA transposons, and the mechanism of their transposition is similar to those of bacteria. The first transposable element discovered by M. Clintock in maize was now known to be DNA transposons. However, the first DNA transposon to be molecularly characterized were the P elements in Drosophila. So P elements in Drosophila. P element present in Drosophila resembles to insertion sequence IS element of bacteria, which possesses a short inverted terminal repeat at both ends and a single open reading frame which encodes single protein transposase. The P element vary in size, ranging from 0.5 to 2.9 kb in length. P elements were discovered by Margaret Kidwell, who were studying hybrid dysgenesis. So dysgenesis in Drosophila, hybrid dysgenesis in Drosophila, dysgenic cross, female lab stock and male wild M cytotype into P cytotype F1, defective hybrid, dysgenic progeny, no offspring. But reciprocal cross, female wild P cytotype, male lab stock M cytotype, produce F1 normal flies. Female lab stock with M cytotype produce no offspring. Okay. In hybrid dysgenesis, a cross between a female from laboratory stock and a wild male yields defective progeny. A phenomenon known as dysgenesis is present in Drosophila when laboratory reared female flies were crossed with wild male flies. In such cross, the laboratory stock are said to possess an M cytotype cell type, and the natural stock is said to possess P cytotype. Laboratory flies do not have P element. In such cross, M female into P male, the F1 progeny show a range of surprising phenotype characteristics in germline, including sterility, high mutation rate, and high frequency of chromosomal aberration and non-distinction. The F1 hybrid progeny are biologically deficient and known as dysgenesis. The phenomenon is called dysgenesis. However, the reciprocal cross between P female and M male, no dysgenesis is observed in F1 progeny. To explain the phenomenon of dysgenesis, a simple answer is that P element transposition. In non-adult P cytotype flies, they possess P element which contain a transposase gene as silence. However, the genes are activated in the F1 generation, resulting in dysgenesis when crossed M with P, female with male. And the cross between M cytotype female flies, no P element, into P cytotype male flies, P element. Each parent contribute respective gametes with respective cytotype. Male gametes provide P elements in the newly formed zygote, which is in silencing free environment, since female gamete is M cytotype. The P elements derived from the male genome can now transpose toward the zygote genome, causing a variety of damage as they insert into genes, and our F1 progeny developed from such zygote are dysgenic. On the other hand, the reciprocal cross between P cytotype P into M cytotype, no dysgenesis is observed. This is because in this case, female gamete provide P element in a newly formed zygote, but the P element is already silenced in female gamete due to the presence of some component in cytoplasm of female gamete. Therefore, zygote developed from such gamete will already silence P element, prevent dysgenesis. So, insertion of P element cause the mutation underlying hybrid dysgenesis. M cytotype shown, then P cytotype shown, P generation, no P elements, embryonic germline cell, no transposase gene silencing, mobilization by P transposase, P element, same cell a little later in time. F1 generation, development to adulthood, then carries new insertional mutagens in germline, flies are sterile.